On this episode of Translating Proteomics, host Parag Malik discusses the importance and challenges of proteoform research with special guest and Northwestern Professor Neil Kelleher. Their conversation covers what proteoforms are, examples of the importance of proteoforms, the scale of and technological advances needed to meet the challenges of proteoform biology. To get the conversation started and introduce Neil, here are your hosts, Parag Malik and Andreas Humer of Nautilus Biotechnology.
On this episode of Translating Proteomics, we revisit a particularly important topic in the world of proteomics, proteoforms. As a reminder, proteoforms are the single molecule variants of gene-encoded proteins. Each proteoform is defined by its full set of modifications regardless of source. And proteoforms are the versions of proteins that we actually find in biological systems. To get a better understanding of proteoforms and their importance, I had the pleasure of speaking with Professor Neil Kelleher of Northwestern University. Neil is the preeminent scholar on proteoforms. He literally coined the term. And I'm very excited to share our conversation covering concrete examples of the importance of proteoforms, the scale of proteoform biology, and also how we can make proteoforms a routine part of biological inquiry.
So maybe this is a timely conversation because proteoforms seem to have their moment. There's new technology evolving that specifically allows us to measure proteoforms. So I'm looking very much forward to the conversation with Neil.
It was really fun. Take a listen.
As you've just heard, I am very excited to have the chance to chat with someone who is the world expert on proteoforms, Neil Kelleher. Neil is the Walter and Mary E. Glass Professor of Molecular Biosciences, Chemistry and Medicine at Northwestern University. He did his graduate work with Tig Begley and Fred McClafferty at Cornell, and his postdoc with Christopher Walsh at Harvard Medical School. His training in mass spectrometry and enzymology really drove his research program of hundreds of publications and a tremendous focus on top-down proteomics, creating the Consortium for Top-Down Proteomics and initiating the Human Proteoform Project. So Neil, on a previous episode, my colleague Andreas and I discussed proteoforms in a broad but somewhat theoretical sense. To start this conversation, I'd like to hear from you directly. What are proteoforms?
Yeah, Parag, thanks, and great to be with you on your podcast. And so proteoforms are it's just basic biochemistry of protein molecules. And it is the mature molecular product from a gene that gets made in our bodies into proteins all the time. So it just means to indicate the complete composition of that molecule. So it would involve decorations to proteins and some other technical words that we'll get into, but it's just the complete compositional molecule of what we call colloquially a protein.
And so let's go back to 2013. You and Lloyd Smith coined the term proteoform. And can you walk me through what led to creating this word? What was the gap that you were seeing?
Yeah, so the word is also a movement. There's a lot behind it, and a lot of people feel strongly about words and semantics. And the word came out of a conference in 2012 we had in Lisbon, Portugal. Drove up the coast, and there was maybe 40 or so participants, and the vendors supported us getting together and talking about, okay, so what are we going to call these things? There were isoforms and protein variants, and oh yeah, there's that one with that decoration and that phosphorylation, methylation, acetylation, all these different forms. Oh, and mutations, genetic changes to protein coding regions. Those are also a thing. And so that word proteoform evolved. And once it was like a moment of magic in the room when that sort of hit. There were different forms of proteins, and you can imagine the moment.
And did you feel that those other terms to describe proteins and their combination of genetic variants and splice variants and post-translational modifications that you really needed to capture that incredible diversity, and it was just not being sufficiently well described in the community at the time.
That's right. There were individual pieces of it, but a single term, and in fact, that's what it came out in the 2013 paper that you kind of gently mentioned, that a single term is in the title, and it just encapsulated all the sources of compositional variation that could occur in a protein. And since then, there's been edge cases and things that we've worked together as a consortium to parse. But yeah, that word now is a thing. And if we're ever going to get to regularizing the proteome, that is to more regularly handle, we're going to have to get proteoforms under our belt. And first things first, what do we call it? And now we have 5 levels that describe different quality levels of proteoforms in the underlying analytical techniques to discover and measure proteoforms.
So tell me a little bit more about that. So the 5 different layers, I'm not familiar with that. What does that? Walk me through that.
Yeah, just a few years ago, like a level 1 proteoform is perfect. You know all of the atoms that comprise that molecule, and you know that the phosphorylation is exactly at serine 720, and you know it starts at this methionine at position 1, and you know the amino acid at the very end of the C terminus, it's called, of the protein, and everything in between. And you know, oh, it's got that mutation is coding, and it changed a threonine to a tyrosine, for example. And all of the splicing that occurs, so the underlying base sequence is basically all the atoms are, that's a perfect level 1 proteoform. Level 2 could be, well, I don't actually know that the phosphorylation is that serine, say at position 100, or it could be at position 108. That ambiguity creates a type of level 2 proteoform, and then it goes on down in terms of quality from there.
I see. And so tell me a little bit, maybe coming up to a slightly higher scale, why are proteoforms important? Why should we care about them?
It's just basic biochemistry that we just want to understand exactly the molecules that are in our bodies. We've done it for DNA and for the most part, and now we need to do it for our proteins because we're built of proteins. They are funded. This is a fundamental concept that is, surprisingly difficult to communicate outward, but it's just so foundational that, we're going to have to confront this at some point. Yeah.
It's so interesting that you say that, because I've seen in the field people confusing post-translational modifications with proteoforms.
Yeah.
And so I'd love to get your thoughts on, first of all, why does that happen? Why are people using this term proteoform to talk about post-translational modifications? And simply, how are they different from each other?
I totally get your vibe right there. It's like, why do I have to go this 2-step dance to get to PTMs that matter? Why do I have to measure proteoforms to get to PTMs when can't I just go after the PTMs? Yes, and the world has done that for the last decades, trying to find the PTMs that matter. But yet we don't have the proteoform perspective. It's like a bird's eye perspective of a particular protein, and that term protein, it's just too simple to capture our biology. That's why proteoforms matter, and that's why you have to measure them to find the PTMs and protein isoforms that matter.
Yeah.
And the argument that is a long logic chain to get through that, but it's just true.
I'm going to come back to that in a moment, just because one of the things that I always think about is that the PTM is this amino acid modified or not? And the proteoform is really the combinatorial explosion on top of that. that says I have 3 different post-translational modifications present on the same molecule, or a combination of a splice variant and multiple post-translational modifications. So it's really that pattern beyond any single site that defines what makes a proteoform.
Yeah, and if you think about it, yes, and let me build on that. If you had 3 phosphorylations on a specific isoform, and that was the particular drug target to really shut down a signaling cascade that's gone crazy in my cancer. Like, okay, I have 3 phosphos in combination, and if you imagine that basically elevates you above the noise band. If that is actually the molecule that exists, you'll see it at the proteoform level. But if you try and piece together those 4 things you threw out, an isoform specific with 1, 2, 3 phosphos, all in, which is actually what we just measured in a protein called MEK1 in a model of melanoma. Sorry, geeked out there, couldn't help it.
Perfect. That's like I said, we're going to dive into that example and more in just a second.
Cool. So that is what I've experienced in my career, is like, oh my gosh, like this is really, 20 years ago, this is hard to measure, but though the biology is there, the function assignment, the biomarker relevance, because biological noise frustrates measurement science all the time. So this is why proteoforms matter, and why year by year this will just continue to grow.
Somebody said there are no proteins, there are only proteoforms, and I'd just like to get your hot take on this. What does that mean to you? Do you believe that?
Yeah, hot take on that, like proteins. I heard someone else say proteins are a myth, that they don't actually exist, that they actually have proteoform. I would say the more complex organisms that, like, so for example, us versus E. coli, you can get an E. coli gene that, will trim the N-terminus and create 1 dominant or 2 dominant proteoforms. And that's just like the central dogma. And it doesn't get altered very much or at high abundance. Okay, but when it comes to humans, I'm then more aligned with your statement. So my hot take is, in our biology, we have this escape from the central dogma, where throughout evolution, and we have a proteoform program of life, it has evolved to constrict the creation and regulation of proteoforms. And yes, there's a noise band. Yes, it goes awry in disease, but there is this dominant canonical proteoform program that we do not have the tools to read out yet. But like you, I think we are on the precipice, within less than a decade. I think we can reach this new plateau where we have tools that match the scale of our biology at the protein in proteoform level.
So I think that's a really great segue. I'd like to come back to a little bit of your history. What was the very first time, what was the very first protein that you saw a proteoform acting differently than a collection of PTMs?
Yeah, that would be on histone H4. Histone H4 through the cell cycle. That really showed me, like, okay, a PTM like lysine 20 on this protein, histone H4. Histones pack DNA into the nucleus, and people around that time were all talking about the histone code, and that multiple PTMs were combining and were actually the algorithm of life, and that lysine 20 on histone H4 gets methylated in, 1 methyl, 2 methyls, or 3 methyls added to this lysine. But how does that occur in combination with acetylations elsewhere or a phosphorylation elsewhere? And so this was quite open. And, just from doing this kind of bird's eye perspective and then lining up 10 different proteoform snapshots from the cell cycle, you can just see this progressive methylation happen through the cell cycle. And I was like, oh, well, that's the logic right there. And the acetyls don't really have much to say about that progression.
So that there's a distinctive pattern over time where it starts off with one modification of one type at one position and then adds on a second one at a different site and sort of stacks up from one to multiple modifications, ultimately defining the behavior of the histones.
Yeah, and there is other biological states where the acetylations matter, like if you're inhibiting with a cancer drug, deacetylation, here we come with the proteoform dynamics, then reveal how much those acetyls build up because you're inhibiting an enzyme that should be taking them off.
That's great. That's great. Tell me a little bit about a few other examples that people may not be as aware of. So you mentioned MAC1, for instance, or troponin or PSA. What are the most glaring important examples that we've seen where proteoforms either drive biology or are better biomarkers or are better drug targets?
Wow, yeah, that's a great wind up pitch, and let me try and hit three fastballs coming at me. And so, but one is PSA. You mentioned prostate specific antigen, PSA. It's generally known, the New York Times wrote about it years ago, that it's a really bad biomarker, that, PSA, the protein. We know there's over 100 proteoforms of this sucker, right?
Wow.
There's glycans that get attached, there's polymorphisms that change the amino acid sequence person to person, and so no wonder that it's a bad biomarker. You're just looking at the level of 80 to 150 different molecules, and you're looking at the total level. We need to look at the levels in prostate patients. So line up 100 men in stage 1 through 4 prostate cancer, line them up, get the technology lined up. This is actually a $3 or $4 million study I'd love to do, and it hasn't been done yet. But the technology is there to do it today, and it wasn't 5 years ago. So that's an example of, like, okay, maybe 5 of those proteoforms of PSA are really specific to stage of cancer. That's the kind of thing that has to be done in the biomarker diagnostic space.
So that sounds a little bit like a hypothesis, Neil, as opposed to a place where we have evidence that says, hey, PSAs that have these 3 modifications are cancer specific, and PSAs that have those N modifications are just evidence of enlarged prostate or something that is not prostate cancer. Do we have an example that we can point at today to say this proteoform or this set of proteoforms is better?
Yeah, so that makes me think about the best business and science case that is the most mature, right, as we wake up today in 2024. It is the biotyper, and this is a proteoform detection engine that's now in thousands of hospitals to tell you very quickly, very cheaply, what species of bacteria is in your infection. And so that's the case and it would not work with small peptide surrogates or these little pieces of proteins. It really has to function at the entire proteoform level. I revert from.
So what is biotyper? What is it measuring in particular?
Yeah, it's measuring about 50 ribosomes are what make proteins. It measures about 50 ribosome proteoforms from a bacterial species, from a bacteria that was grown for a few hours in a dish from an infection. That's the assay. And it was launched by Bruker Daltonics many years ago. But it kind of proves the business and science case for those who want a more mature example.
Yes.
There, we have in more complex disease, so detecting a bug that's in a person, okay, that maybe is a little easier than, okay, now I have cancer, or I have another example I'll give you is in transplant, in transplanting organs. We have a panel of 24 proteoforms. And so there's a few proteoforms of chemokines. This is in PB, these are in the white blood cells, the immune cells of your body, which are the first to react and reject a transplanted liver in this case.
So, Neil, these are great examples of a biomarker and in the case of histones, something that is driving biological function. Are there other places where, when you think of proteoforms, that if we don't understand proteoforms, if we can't characterize them, we're limited?
Yeah, the troponins are a great example. In cardiology, the work of Yinga in Wisconsin, she really clearly showed that as your heart, as you get enlargement of the heart in pig models and in humans, that you have this protein troponin that has just a simple story, 3 proteoforms, and there's 2 serines right next door to each other that go pop pop, and that's what happens in healthy biology. That is the healthy proteoform program running in my chest. That's what you want. And then as you get enlargement of the heart, that phosphorylation, those phosphoproteoforms go down.
Mm-hmm.
And to an alarming degree in very enlarged hearts. And so that actually tracks progression of disease. It's quantitative. Now that you know that, you then could go back to other types of assays and build assays that are proteoform informed.
Yes.
And so there's a distinction between, okay, how do we discover this biology, which in retrospect is really clear, the signal's really clear at the proteoform level, versus proteoform resequencing or proteoform informed biology. And that distinction we could get into, but I think it's just it's that bird's eye perspective of just what's going on to your proteins.
So I think this is a great opportunity to talk about the goal of building a proteoform atlas and a human proteoform project. So this is something that you've been driving with support throughout the community. Tell me a little bit about the goals of this project and what are you hoping to achieve with it?
Yeah, Parag, the Human Proteoform Project is, as we just described, something like 5,000 cell types and determining the proteoform landscape in each cell type. And of course, that means different organs, like the kidney would have something like 80 cell types. And we would determine it at a given depth, equivalent conceptually to the 4 or 5x coverage. So you'll recall the Human Genome Project had a 4x coverage when it was first done. Now it's 40 or 50x coverage, because look what happens after you regularize a biomolecular landscape in the human body and create all these economies of scale after it. That's what the project is meant to do. But the project itself is 5,000 cell types, determine the proteoform landscape in each kind of cell, plus the 12 fluids of the human body. And after that, you would
I
have.
don't think I realized there were 12 fluids in the human body. So
there's a lot of side fluids, and some of them your listeners may not wish to hear all 12 body fluids, but
fair enough. Let's not go into that.
Yeah, that's the atlas of the Human Proteoform Project would create, along with the technologies that finally would at least regularize, domesticate, sequence the proteome, and to make it not so enigmatic that we as a species, we domesticated plants 10,000 years ago. We can do this as well. And after all, precision medicine 2.0, it really requires us to confront the complexities and harness the proteome, which right now is enigmatic and open-ended and just makes drug discovery and diagnostics just really challenging. And also for the business sector, it's really difficult to establish drugs and do so with anything more than 5% success rate all the way through clinical trials. So this is what the project is meant to do: level up our understanding of ourselves at the protein level.
Great. So Neil, I'd like to ask two quick follow-ups to that. One is the project that you've just described sounds immense. Is this 100 years of work? What is the scale that we're talking about to complete the Human Proteoform project?
Yes, that's a great question. And the Genome Project also seemed quite challenging in 1991 or 1990 when people were talking about it, 3.1 billion bases. How are we going to possibly sequence that? Within 10 years, it was done. And that's what happens when you bring the best minds in the world to the problem, and the belief that it can be done. That's not to say that it is not immense. It's a significant challenge. You're talking about doing a project, but not until, and this is where the private sector conceptualization gets involved. The Genome Project didn't go to production scale until it was $1 per base. And that was 1994, 95, really. And so $1 per proteoform is the price threshold. And at that threshold for defining the reference set of human proteoforms, that's not to say all in the universe, you're looking at a same arc and scope as the Genome Project. So you're looking at 10 years and something on the order of 3 to 4 billion US dollars.
That seems very tractable. So I'd love to double click for a moment on the question, how many proteoforms are there? Because one can imagine there's a protein like tau that has 85 phosphorylations. There could be
it's so complicated, Parag.
2 to the 85 possible combinations of just those. And that is a terrifyingly large number.
Yeah, back when we were talking about the example that had one isoform due to a splice variation and then 3 phosphos,
Yes.
and all those things occurred in combination. And then I jumped on that and I said, ooh, there's a filtration there that it's not that biology populates all the possible proteoforms in the universe. No. There's a great constriction of proteoform explosion that could happen, or that can happen in someone's computer on their desktop, but doesn't happen in the body. And your example of tau with 65 phosphos and 6 isoforms and all, you could mathematically create 6 times 2 to the 65 power. Oh my God, it's so complicated. That's why I reacted, because it's different than a lot of the ways people react to that complexity. And I will say that the tau proteoform landscape, if you react the way a computer would and propose all of them or explode them, you say it is too complicated. Because 2 to the 65 is a ginormous number.
Yes.
But the biology doesn't create all those combinatorial events. Why would it? If you had that much leakage of ATP and the kinases were dropping phosphos on, that's an enormous waste of energy. Therefore, we have a proteoform program of life that makes a constriction a reality, lowering the number of proteoforms, allowing us to actually create tools to domesticate, regularize, and deep sequence the proteoform landscapes in our body. And tau is a great first example. But I can expand more on that, like how many proteoforms are there? Yeah.
I would love to hear, because we had a paper that asserted there were about 6 million proteoforms, and we've just established that tau alone could have more than 6 million proteoforms. So how do we get from our current state of knowledge to
Yeah.
an estimate of 6 million?
So that would be from an entire cell, like a single cell sitting there in and doing its proteoform program. And 6 million is a good estimate of, if you were developing technology to determine the proteoforms in a cell or a type of cell, a kind of cell, like maybe you needed 10,000 or 100,000 of those cells of the same type, you could set a threshold of, say, 6 million or 1 million proteoforms that you would want to sequence in such a cell type. Now, it just so happens that the world is creating an ontology or a known set of all human cell types. And that number is something people still quibble, but it's, something around 5,000 different kinds of cells that make up our body. So for example, in the kidney, you have, looks like approaching around 80 different cell types. So you would want to know in a deep human proteoform project, the 80 cell types. A deep version would be 6 million in each cell type.
Oh,
That
I
would
see.
be.
So it's not 6 million in the universe.
Mhm.
So the universe of proteoforms might be much larger than that, but within a given type of cell, about 6 million.
And the 6 million is a deep version. I think a million is a good doable number. And let me also make one other, pro tip, expert moment. There are redundant proteoforms, like the form of histone H4 that I mentioned earlier,
Mhm.
that has a dimethylation at lysine 20 and an acetylation at lysine 16. And oh yeah, an acetylation at the clipped N terminus. Okay, that proteoform, it's going to exist in many different types of cells. So then it causes you to have to say, oh, do you mean unique proteoforms, like a new molecule added to the atlas of all proteoforms in the human um, uh, body that we wish to measure at a defined threshold? Or do you mean redundant or unique proteoforms? Those are two different counts, but I think the number you're after is about 100 million in a deep version of a project. 100 million proteoforms, unique proteoforms, would be a deep scale of, it'd be the equivalent of the Human Genome Project, but at the
Okay.
proteoform level. Yeah.
Maybe just one follow-up question there. You mentioned that one of the things that's going to have to happen technologically in order to enable a project of this scale is bringing down the cost of discovery.
Mhm.
What are the other things that need to change, the other holes in technology that are required to bring proteoform analysis into routine use?
So there you go with the chicken and the egg question. That's perfect, right? Because without the reference genome, the genomics and next gen genomics would not have flourished at the time points, into 2000, 2002 to 2007. You could have next gen sequencing emerge because of the role of government and its leadership role that they grabbed onto. And so it's very similar here, that proteoform discovery itself, which I think your question was about the discovery part.
It was about discovery. So just starting with discovery, what are the barriers to discovery that we need to overcome?
Yeah, it's about 100-fold off in cost and speed of where it needs to be. If those things could be addressed, we would have the tech to do the project, which is actually less of a jump than the Genome Project had if you go back to 1991 through to 2000. And yet we have a more challenging issue that we can't replicate. You know, we don't have PCR or the way to replicate and amplify DNA proteins. We don't have that. So yeah, that's what's required is a platform that can emerge to do it at $1 per proteoform for discovery. Resequencing of known proteoforms, that is significantly less expensive even today. And there's new technologies on the horizon that you're well aware of that are well positioned to do that resequencing. The discovery part of it, you're right. Yep, it needs to be 100-fold drop in cost and 100-fold increase in the speed and throughput.
What is the barrier, either sociologically, perceptually, between where we are today, where frankly most biologists haven't even heard the word proteoform,
right,
to that future world where everything is done with proteoforms?
This is, I think, in decades, you first find a way to move the project forward that has use cases. You remember in the Genome Project, you had basically genetic mapping. There was this stage of where they created a genetic map. And so what were they doing? They got families together that were all stricken with the disease, and they were sampling those people. So early on, people were like, oh, this is directly around disease, right? This isn't, that's what the project was. But then it went on to a different kind of mapping. I think this is where we're at, that I think brain diseases, neurodegeneration, it really is unfortunate that there is technology in the world that could lead us to really show use cases, turning them to success cases in neurodegeneration, and do that with urgency. Because right now, most of the resources are not proteoform informed in neurodegeneration. And I will stake a very large amount of my personal wealth on that, which is not very much, on that bet, because proteoforms will shine. And I think that would then go, oh, yes, we need to do this across all human biology. That's one strategy to try to do that. But that itself is a $50–100 million effort to really do that at scale. Otherwise, it's going to be a slow burn until we, every year by year, we're getting closer. It's making more sense to more people year by year. And that's all I can promise your listeners, is that the more that you drill down or double click into this, the more sense that it makes.
That's great. I think from my perspective, it really is about getting a few very clear examples where the protein level alone, the PTM alone was insufficient. And if we look at, for instance, immunotherapy, it was for decades being worked on. You hear Carl June talk about his journey where people thought it was crazy talk and he had trouble getting funding. But then there was a moment where a series of independent studies came together and said, wow, this is notably different and better than what we saw before. And I think proteoforms just hasn't had that use case that is so clear and so simple and understandable by every biologist. I also think accessibility is a big part. So I think if your lab comes up with the use case and you're the only lab in the world that can do it, it's simply not going to penetrate all of biology. It needs to be something that is accessible to everybody alongside compelling.
Yeah, and what are our options, right? Where it's the chicken and the egg problem, where every technological plateau that you want to level up to, has this chicken and the egg problem. And yeah, it is a challenging measurement for the last 20 years. It's been challenging to make the measurements. And so like our prostate specific antigen, the PSA test for prostate cancer in men, classic example. where I can see exactly what needs to be done. There is technology in the world that can do it, but you've got to get the patient cohorts together. You've got to get the sampling done. You've got to then do the study and then report it out. And that's a 5-year, $5 million, $4 million effort right there, times all the other use cases you would want to show. And the tech just isn't widespread yet. That very pocketed, very siloed in certain labs.
Well, that's a great opportunity for the world, is to first work on those demonstrations, and then in parallel work on the technologies that can democratize access to proteoforms, not just proteins.
But yeah, I think just on basic foundational fundamental grounds, first principles, it's that simple. It's just you want to know the exact molecule that is produced in my body.
Yeah.
And I think that argument, if people can slow down enough in a very busy world and a big marketplace of ideas to just confront that and all of the transformational things that would occur were you to able to have the technology that matches the biology in our bodies.
Fantastic. Thank you so much, Neil. This has been incredibly informative. I really appreciate you taking the time to chat today.
Parag, thanks for the chance. So I always want to talk about proteoforms, and good luck to us in the future.
So Andreas, I have my own ideas, but what would you say is your key takeaway from the conversation?
So I stuck my head out and publicly went at Hoopo on the podium and said, there's no proteins really, it's only proteoforms.
Right, and I think you might have stolen that from somebody.
Possibly, but I think it's worthwhile a serious conversation.
Yeah, so I think proteoforms are clearly important. But I also think that we really just don't have enough information yet. We have a couple really clear examples of where proteoforms matter. But on the other hand, we have tens of thousands of proteins we believe there are proteoforms for, but what they actually do, how they function, how they drive biology, to me, it's just completely unknown so far.
And I think portion of what fascinates me in the conversation with Neil is that he termed the technologies we have today as proteome unaware. And I think what we need to focus on moving forward is we need to create more technologies and use technologies that are proteoform aware. And I think that's a critical next step for the research community.
I think that makes sense because, for instance, if I look at a band on a gel, it just tells me the mass of a protein. It hides a lot of detail that I might see in a Western, where I see phosphorylation, for instance. And the band on a gel is still useful, but if I know that there are 6 different phosphorylations, that gives me a little bit more information and allows me to interpret and understand what's going on. And I sense that's what you're saying with proteoforms as well.
Yes, and it's not the fact, it's a fact that we have been able to observe proteoforms. 2D gels are a good example. It was just very difficult to actually understand the individual proteoform that actually is being separated on these 2D gel spots. And so
I think what you're saying is that 2D gels are terrible.
2D gels are terrible in that sense that they give us a lot of information that we can interpret because we really don't understand what is in the spot itself. So I think it goes back to we need technologies that really are aware of proteoforms and able to resolve these complex structures.
Okay. So how do we think about the data that's been collected, the decades of research that's been done with 2D gels and Westerns and other, where we didn't have a knowledge of the proteoforms?
So I think some of them, results will still be valid. And I think in many cases, the results will be, updated to include the perspective of proteoforms, right? I mean, part of the conversation with Neil is how big is that space? Yeah. And, if you just theoretically speculate about it, it's astronomically big. Yeah. I view this from a perspective of there's potential that every cell has a different proteome. What's more important, though, with respect to the number is can our organism, particularly humans, create sufficient proteoforms to actually adapt to the environment? So it's not the total number that's important, it's the total possible combinations that can actually help us adapt to the environment.
So it's really about flexibility.
Yeah.
It's about to recognize, to deal with the unknown, and the cell having a lot of levers available to do that.
Correct. And I think the challenge is going to be, because there's so many proteoforms, where do you stop?
Yeah.
And do you have the technology to find the ones that are relevant?
Yeah, I guess my perspective is a little more nuanced than that. I think that there are sets of modifications that are really just there for mechanical purposes. So if I have a giant glycosylation on something, another protein maybe can't approach because there's a giant blockade on that protein. And whether that glycosylation is on site 1 or site 2 or site 3, maybe it doesn't matter as much. On the other hand, there may be other proteoforms which are defined by very narrow changes in the charge at a site. And that can, whether it's at that site or a site that's further away, may be incredibly important. And that's our challenge, is how can we take this landscape that is so infinitely large and differentiate the things that are functionally important from the ones that are just kind of there.
So do you think we're going to be able to have proteoform-aware technologies in the next 5 to 10 years?
Oh, I absolutely. In fact, I think much sooner than that. I think technologies are emerging today that are so exciting and are going to allow us to unpack all of this complexity and really understand how proteoforms drive biology.
I'm looking forward to that.
Thanks so much for joining us on this episode of Translating Proteomics. Today we had a vibrant conversation with Neil Kelleher, learning about proteoforms, their history, their impact, their importance. And I'd love to hear from you. Have you ever heard of proteoforms before this episode? What do you think? How important are they? Where have you seen them in your life? That the best explanation for the biology you saw came from a proteoform? Chat with us in the comments or send us an email. We'd love to hear from you.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.